like for this typically depend on the traits, single gene. If it's recessive, if you want to select the trait, you have to you self-cross at each back cross generation. So it takes like many years, 5-6 years to achieve this goal. But in the marker selection, back crossing, it's become much straightforward. We don't select the trait phenotype itself. Instead, we use the foreground selection and background selection using molecular markers, which can very efficiently and quickly recover the recurrent apparent background, but at the same time select the target gene. So back markers and the back crossing is very, very effective. It's also a very good tool for gene pyramiding for multiple disease resistance. So there are many advantages for marker assisted selection. We can combine with foreground selection. Excuse me, I feel a little bit. Can I take a break for 5 minutes?
Oh, okay, no problem, sir.
Sorry for this. Let me take a 5-minute break.
Okay, sir.
సార్ ఫైనల్ మనం చెప్తాను తర్వాత ఇది కరెక్షన్ చేయాలి ఫొటోస్ కూడా తీయాలి ఫొటోస్ తీయాలి నేను వస్తాను క్లోజ్
Sorry, can I resume?
Yes, sir. Yes, sir. Please start, sir.
Yeah, it's midnight. I don't feel good today, so I'm really sorry for this. So I'm going to continue. So
Yeah.
there are many advantages for marker-assisted breeding. First, you can do background selection and foreground selection, which quickly recovers the recurrent parent. You can select heterozygotes, which is particularly true when the target gene is recessive. So you don't have to do self each generation to see the phenotype. You just select the allele of the target gene. So you don't really need to, for these resistance, really need to test these resistance each generation. So, which is, often affected by environmental factors or spend a lot of time, money to do the test for these resistance of quality. And you can also select earlier, often at the seedling stage, you can do the selection, only keep those you need. So that's where economically very significantly save money. In particular, you can select multiple genes at QT at the same time. So it's much faster, more accurate, save time and save money. Of course, we cannot do without necessary, there are some requirements for doing practicing marker selection. So you need the necessary genetic genomic resources like genome sequences and molecular markers, genetic maps, and also need the genotyping platform. And for the trait, you must have a very good understanding of the genetics of your target traits, like disease resistance, fruit quality, plant architecture, etc. So you need to know how many genes control the trait, QTO, what's the inheritance mode, in fact, are there any interactions or any background infected, etc. And you need to establish marker trait associations, which is a prerequisite for marker selection. So when you talk about a cucumber, we have over the last decade, we have developed the cucumber genetic genome resources, significant increase of this. For example, we have at least 3 cucumber lines with draft genome available, and particularly now we have more than 10 at this time. This is a GY14 pickling cucumber. We developed a draft genome 10 years ago. And from this gene assembly, we were able to develop high resolution genetic maps. Several genetic maps have been developed. Here is an example. Another example with 11,000 SNP loci, a genetic map we developed. And we also recently improved the draft genome for GY14. We have a 2.0 version 2.0, which has been much improved compared with the first version. This draft genome assembly is publicly available at the cucurbitagenomics.org website. Everybody can access and use it. So compared with another called 9930, we have increased about almost 50 MB bases of the genome assembly, the GY14.
అంటే ఒక ప్లగ్ బోర్డ్ కి ఇచ్చేయండి ఏదైనా ఒక ప్లగ్ బోర్డ్ ఎక్కడైనా ఏ రూమ్ లో
over the last 10 years, the number of genetic studies identify genes, molecular markers for targeted traits, and this example, some disease-resistant genes or QTOs which have been mapped, cloned. Last couple of years, there are several recent reviews about the genes and QTO with molecular markers in cucumber. If you are a cucumber reader, you should have a better understanding what kind of traits, what kind of molecular markers we have for particular traits, which could be used for marker-assisted breeding. Here is an example for the how molecular markers were developed through QTO mapping, like for Passana-Capi, for low temperature germination capacity, for these resistances. Here, example, we through QTO mapping, we identified several QTOs for dominant mutual resistance in 2 PI lines. Some major QTOs we already cloned it. This kind of information is very important for their use in breeding these resistance. My last topic is about the use of crop-wide relatives. As I mentioned, the Cucumis hystrix is the only wild species, a secondary gene pool, which we can use for, which is actually compatible with cucumber. That means we can use some of the traits. Indeed, there are some unique traits which are not found in cucumber, like root nematode. There are some unique, like dominant mutual resistance, virus resistance. So unfortunately, Cucumis hystrix have 24 chromosomes, while cucumber has 14 chromosomes. So we cannot directly use it. We have to do into specific crossing, and then to induce, to synthesize artificial tetraploid, which will be used as a bridge to introduce genes from hystrix to cucumber by development of introgression libraries. So in the past, we have successfully developed into specific hybrids between cucumber and hystrix using embryo rescue. These pictures show how we do embryo rescue by basically take the hybrid embryos onto culture media and grow them into plants, and then using colchicine to do induce chromosome doubling for the artificial tetraploid. And then the tetraploid can be used to cross with cucumber to bring the chromatin into cucumber background. Of course, for successful use of the alien genes, you must have genetic recombination between chromosomes from hystrix and cucumber. Yes, we use cytogenetic studies to provide evidence for the genetic recombination between hystrix chromosome and the cucumber chromosomes. So over the last, we have been working on this project for several years, and we have developed a series of introgression lines which carries different fragments of DNA pieces from hystrix, and we are trying to evaluate if any introgression lines have any unique traits which can be used for cucumber improvement. So these are what I might talk today, and lastly, in the last 10 years, there are many people come to the lab and help to do various traits achievements. So these are some pictures of lab members from different time, different year. Yeah, I'm fortunate to have many people to contribute to this program, including some from Dr. Pradi Kumar T. I think some of you may know him from Kerala. Dr. Mamata from Punjab University in these pictures. So we have also from India Institute of Agricultural Research. So I really appreciated their contributions over the years. So that's all I have for today's talk, and thank you for your attention. I will be happy to take any questions if you have. Thank you.
Thank you. Thank you very much, sir. You have covered all topics you have covered, especially in cucumber and the classification, hydroponics cultivation, genetic diversity, types of different cucumbers, biodiversity, molecular markers, and the structural changes in chromosomes during domestication, traits under domestication, and the bitterness causes, breeding goals for pickling cucumber, research priorities, trends in cucumber breeding, marker-assisted selection and backcrossing, cucumber genetic map, marker-associated traits, methods of breeding, hybridization, and polyploidy with colchicine. So, sir, everything you have covered, it is very excellent. Thank you very much, sir. And a very, very nice lecture given by you. Thank you very much. And now it is open for the discussion. Any question and answers can all the participants can ask the sir. Please. Our Director of Research, Dr. R.S. Kirit sir, also there. Sir, any questions from the students, scientists?
Dr. Dhananjay, I had the opportunity of hearing his lecture physically when he has given this particular work that was conducted, especially in gene pyramiding in case of vegetables. When I have gone to IIVR, Indian Institute of Vegetable Research, there he was there, the same lecture was given there, and something more was added here in this particular lecture. I'm very happy that you have invited for this particular Global Connect series. So it is very comprehensive, and Dr. Weng is one of the important contributors as far as the complete mapping of cucumber genome. Actually, this is one. That's what I wanted to say here.
Thank you. Yes, I'm lucky to have several
I was lucky,
very
sir,
nice,
actually.
excellent scientists to visit my lab, contribute to
Thank you very much, sir.
work I reported. Yes, I still have some collaboration with some colleagues in India Institute of
because now we have
Research
established that in our university, we have established the facility for marker-assisted selection, sir, especially. So this is where your guidance is needed for our students' research, especially. Thank you very much, sir, for coming.
You're welcome. Thank you. Yes.
Addressing our the students as well as the scientists of our university.
Yeah, feel free if you have any questions, you can send me email too. Anytime, I will be more than happy to answer.
Dr. Gangadhar, are you there?
Yes, sir.
Yeah, it is very comprehensive.
Yes,
like
sir.
Dr. Weng, I think you can interact. So where secondary gene pool is also exploited, you have seen.
That question and answers. Any? Students, scientists.
Good evening, Doctor. Hello, am I audible, sir?
Audible.
Sir, very nice and comprehensive lecture, sir. I see that you have worked on several of the biotic stresses, sir, like several diseases. Is there any success stories from your lab you have worked on using wild relatives of these cucurbits, and you have incorporated those genes into the cultivated varieties? And I would also like to know what are the main biotic constraints in your North American conditions?
Yes, as I mentioned, the Cucumis hystrix is the only wild species we can use, but really the trait we would like to explore from this wild species is rock knot nematode resistance called incognita. Unfortunately, the accession I have for the Cucumis hystrix doesn't seem to have high resistance for the rock knot nematode. So at this time, I have a couple of introgression lines with Donemigio resistance, but we already have good resistance for Donemigio in cultivated cucumbers. So it's not a really high priority or exciting, though from plant breeding perspective, we always went to diversify the genetic base for these resistance. So it's still a very good source of resistance from the Cucumis hystrix. So that's the first question. Second question about the what's the most important issues in cucumber production for biotic stress. As I mentioned, the top priority is Donemigio. Donemigio is an homicide pathogen which really devastating the cucumber production in the U.S. Particularly since 2004, when the old Donemigio resistant gene was broke down. And in the first few years, it really was devastated. So totally rely on fungicide. But now we have these resistance from introduced from some land races into cultivated cucumber, although it only has moderate resistance, but it's still better than nothing. So we are trying to combine different sources of resistance into elite genetic backgrounds to improve that Donemigio resistance. In addition to Donemigio, we have some other issues, particularly like virus resistance for CGMMA, which is particularly a problem for greenhouse production. We are trying to screening for CGMMA resistance. Some other issues, it's not of priority, but still cause some concern, like angular leaf spot, attacked leaf spot passages. But we have good source of resistance though, for those. So I think that Donemigio is a top priority for biotic stress tolerance.
Sir, in
US
Indian conditions, viruses are major constraints, sir. I have worked with CGMMB for developing the disease diagnostics.
Okay.
So I see that many other viruses are also there. So what is the breeding strategies for combating such type of viruses in North American conditions?
Virus, we're still looking for, of course, you see that called integrated pest management, but I'm a breeder and a geneticist. I'm looking for resistance for any pathogens or pests. So for CGMMA, yes, it is an issue. We just started screening, working with a virologist in USDA screening cucumber collections. We are fortunate. That's just started not long, a few months ago. The initial screening where they find some resistance for CGMMA. So we are trying to initiate that genetic mapping identify molecular markers, and they introgress the resistance into elite genetic background.
Okay, sir. Thank you, sir. What is the status of GSB, sir? What is the status of gummy stemplate in your cucurbits in your?
Gummy stem blight is not an issue in US cucumber production. I don't have any experience working with GSB. We do have colleagues working in watermelon gummy stem blight, but I don't have any experience working with in cucumber.
This question was because when we worked on the same disease, Dr. Umesh Reddy, he was telling that in Virginia conditions, it was a major constraint. So I was wondering if it is a major constraint in your Wisconsin area like that.
No, Wisconsin, because the weather is temperature, the summer is cool here. We Porter Mitchell angular spot product two concerns in Wisconsin, pickle cucumber production. No Donny Mitchell, no other viruses. We have a very cool temperature in the summer in the north, Wisconsin.
Thank you so much, sir, for your clarification. And it was very comprehensive lecture. I congratulate you for your delivering a very nice talk, sir.
Thank you.
Thank you, Seshkaran. Any questions from anybody? Sir, actually today our Honorable Vice Chancellor is to participate due to preoccupied work at the secretariat level. Sir went to the government secretariat. So he's very much interested to listen hear our lecture, actually.
Thank you, appreciate.
Sir, any questions, anybody?
Morning, sir. Myself.
Good morning.
I'm working in Parvatipuram, sir.
Okay.
Sir.
Parvatipuram, okay. Ask question.
Sir, have generated QTL population, sir, in related to that bitterness resistant lines? Mapping population have generated, sir?
Madam, your voice is very low.
Yeah,
Speak loudly.
Your volume is low.
Hello,
Speak
sir.
loudly.
Hello.
Repeat the question.
Sir, have generated QTL population, sir, in relation to bitterness resistant genes?
Bitterness resistant genes.
In related to QTL population have generated, sir, mapping population?
You mean what kind of mapping populations for these resistance?
Yes.
It depends on.
Madam, you ask clearly.
It is not audible.
Your question, no clarity.
Okay, sir, I will send message, sir. It is not, signal is not.
I can hear the voice is too low, but so are you asking what kind of populations, mapping populations for disease resistance?
Bitterness resistant lines.
Bitterness resistance.
Bitterness resistant lines in cucumber. What is their performance in field level, sir?
Sir, madam is asking bitterness resistance lines performance at field level.
Yes.
Bitterness, you mean?
Yeah,
Bitterness, oh, sorry. Yeah, for bitterness, as if you read the science paper published, first, the different genes control, some control the biosynthesis of the cucurbitacin chemicals. There are also transcription factors which regulate the biosynthesis of these chemicals in leaves and in the fruit, which is controlled by two different genes for the chemical accumulation in vegetative organs and fruits. However, the expression of these genes is influenced by environmental conditions, particularly temperature, the soil moisture, like a drought condition will affect expression and cause accumulation of the cucurbit gene. So that's why you often see if a cucumber field have very dry drop stress or cool low temperature stress or high temperature stress, the fruit will become bitter. Even the plants carry the non-bitter gene. So this gene expression is affected by environmental conditions.
Mostly the bitterness is affected with the climatical conditions. What you say?
Yes.
Yes.
In regular optimal conditions, they are okay.
Yes, sir. Madam, your doubt is clarified?
Okay, sir. Thank you, sir.
Okay. So any questions?
Sir, do you have any resistant sources for bigoma viruses in cucumber?
Could you please repeat?
Hello, sir.
Hello.
Ramana, repeat it.
Sir, do you have any resistant sources for bigoma viruses in cucumber?
Pickleworm?
Yes, sir.
Pickleworm is not an issue in U.S. cucumber production, but I did read literature. When I started my job, I read the literature. There are a few lines in U.S. cucumber collection which have been shown to be resistant to pickleworm. But I didn't work on the. If you read the literature, cucumber literature, you know this. And also, I think there is a new scientist in USDA areas, Charleston, South Carolina. He is working on pickleworm. He's a new scientist. I haven't had a chance to meet him. So basically, yes, there are some lines which have been shown to be resistant to pickleworm. And there is a person who probably is working on this pickleworm. So that's my answer.
Okay, sir. Okay, thank you,
Sir,
sir.
his question was related to the bigoma virus resistance, sir. His question was related to the resistance to bigoma viruses.
Bigoma virus resistance.
Yeah, his query was related to the resistance for the bigoma viruses infecting cucurbits. I was referring to the previous speaker's question, previous scientist question.
Sir, understood that bigoma virus resistant lines, any lines are there identified at our center?
Sir, bigoma viruses are more recently damaging cucurbits in India, sir. It's spread and incidence is very high, sir, in all the cucurbits. Suppose, do you have any resistant sources for bigoma virus, especially in cucumber or ridge gourd or bottle gourd? So we want to have that material, sir.
Because I don't work with pickleworms. I don't have much knowledge, as I mentioned. I know the literature which were published a long time ago in cucumber. There are a few cucumber lines which have been shown to have resistance to pickleworm in cucumber. And I'm not familiar with other cucurbits.
Sir, it's a bigoma viruses, sir. Sir, it was bigoma viruses that are transmitted by whiteflies. His query is related to bigoma viruses, which Gemini viruses like transmitted by whiteflies. So any resistance
I don't have much knowledge about the virus too. Sorry. Yeah, because I'm primarily focused on passages which are in the northern Midwest area of United States. So viruses have never been an issue in our area.
Ramana,
actually in Indian conditions, sir, these whiteflies are a big menace, and consequently the viruses that are transmitted by those whiteflies.
Yes,
So it
we-
depends
we-
on the
we have-
Indian conditions, sir. Like probably in North American conditions, the whitefly menace is less, and relatedly, like the
No,
viruses that are associated with
it-
them are.
it's an important issue for mainland in California. I know, and also we have people, mainland breeders working on whitefly resistance and virus resistance, but not the cucumber.
Okay. Okay, sir. Thank you. And virus is the major, not problem at, I think, in USA. Here the major problems in India. So any
No, sir,
questions?
in USA also that there
Yeah,
are
it's in California,
eastern US
whitefly.
and western states, Florida and California. There we get tropical conditions. So probably the whitefly incidents and the related viruses might be a major problem there,
Definitely,
un-
definitely. Yeah,
and not under Wisconsin or Virginia states.
Okay.
the Central Valley, Imperial Valley of California is basically incubator for all the viruses and whiteflies. Different biotypes and new strains of virus. Yeah, it's an issue in California.
Sir, any questions? Otherwise, we'll go for vote of thanks for our registrar, Dr. K. Gopal Garu. So any
Yeah.
questions finally?
If there are any questions, Dr. Wang definitely will be kind enough to answer. His mail ID has been given in the flyer. So if somebody wants to have a clarification, they can give directly mail to Dr. Wang and they can have a clarification. Thank you.
Feel free. Thank you.
So Dr. Wang, really it's a great lecture. Definitely our researchers, especially breeders and horticulture people who are working in the breeding aspects, and also the PG and PhD students definitely benefited by your talk. Talk is really exhaustive and very nice. Of course, this Global Connect series program has been started in our university. After arrival of joining of our Honorable Vice Chancellor. So it is the brainchild of our Honorable Vice Chancellor. We thank him for starting this nice one, which is beneficial to all the teachers and also the students. We thank you very much for giving a nice lecture. We thank our Vice Chancellor for starting this nice program. And we thank our Director International Program for arranging the nice Global Connect series lecture. That is your lecture. It is very nice. And we thank all of you. And we thank all the participants who benefited by this, participating in this. Thank you. Thank you very much for making this Global Connect series a grand one. Thank you. Thank you very much.
Thank you.
sir.
Appreciate your invitation, and also thank you all of you spending time to this talk.
Thank you, sir.
Thank you, sir.
Thank you, very nice, sir.
Okay.
now
Have a
we-
great day.
Yeah, okay, thank you, sir. Same
Bye.
here, sir.
It is good morning for you.
Yes, it's 1 a.m. 1 a.m.
Yes, yes, yes.
Bye.
It's almost 12:30 a.m. at your place. Oh, it's around 1 a.m. Sorry, 1 a.m.
Sorry, sir, we disturbed you. Thank you very much.
No,
sir.
it's,
Thank
it's
you,
a,
thank
it's a
you very
nice
much.
experience. Nice experience. So
Thank you.
thank you again for your invitation.
Thank you.
Thank you, sir. Thank
Bye.
you all. Thank you all. Now it is closed.